Okay, thanks. We're multicellular, right? A human body is made up of, I think, billions. It might be trillions. I'm not sure. We certainly have trillions of bacteria in our body. Now, each cell, in each of our cells, we have a nucleus because we're eukaryotic. And in each nucleus, we have the whole genome, all of it, not just part of it. We have 23 pairs of homologous chromosomes. And what that means is, each chromosome comes in 2 pairs. These are identical chromosomes in that they have the same genes on them, but they might have different alleles. One of those we'll say that's mom and that's dad. So we are a diploid organism. That means we have homologous chromosomes, 2. So we have 23 pairs of homologous chromosomes. In other words, we have 46. We have 23 from mama and 23 from papa. And each pair has the same genes on it, but they might have different alleles. So, homologous. Homologous means the same. And then, of course, on each chromosome, there are a whole bunch of different loci or genes. And these code for proteins. And it's like one long DNA molecule, and the genes are functional regions of the DNA, where the DNA just spells out which amino acids to make, and then those instructions are translated into proteins. Okay? And then DNA is a double helix. As you can see. And RNA is only a single strand, but DNA is a double strand. Double strand, it looks like probably some of the first life might have been RNA based, but DNA is much more stable. And there are 4 base pairs. These are nitrogen bases for each gene in terms of amino acids. So each gene has either, they come in pairs, A and T, adenine and thymine, or cytosine and guanine. And if you have 3 of them, that's a codon that codes for an amino acid. So a codon is 3 pairs, 3 bases in a row that code for a specific amino acid. And we'll look closely. Okay, so here's our DNA, right? There's the backbone, and made of sugars and phosphates. And that's along here, right? These are all sugars and phosphates. And then there are these nitrogen bases, and in this case, a base here, and a corresponding base here. Okay? So A always goes with T, and C always goes with G. And the bases are T. Why didn't, oh, I know why. Come on. Oh, for Christ's sakes. Okay, anyway, it'll show up. So that means there are these 4 nucleotides. They go together. A and T go together, and C and G go together. So if I gave you a sequence of nucleotides, let's say I gave you a sequence of CCTA, what would be the complement? Was it?
GGAT.
Yeah, GGAT. Okay, so you should be able to do that. You always know that a T goes with an A C goes with a G. Okay, so here's a complementary pairing of nucleotides with the sugars and phosphates, the whole bit. Now, the reason they pair is because they have different chemical structures, and so if you were to try to put an A and a G together, they would be mismatched. They wouldn't fit. So with C and G, you can see how they're arranged. A has a different look than C in terms of the amine group. And there's a complement. So each strand has the complement and the base pair. There's always 2 base pairs together. All of our chromosomes are actually strung together. It's over 3 billion base pairs. But very few of those base pairs actually code for proteins. It used to be thought that they were junk. There was a lot of junk in them. But things have changed. Okay. In 1953, these 2 guys, there was a race on. People had finally started to figure out that maybe DNA was important. They didn't know what it did. They didn't know how, where genes were. They didn't know how hereditary information is transmitted. And these guys worked out the actual structure of DNA. Watson and Crick. Crick is dead. Watson's still alive. He's the old guy. Crick went on not only to figure out the structure of DNA, but figured out the coding, like what 3 base pair combination codes for which amino acid. So during DNA replication, now not all of this has to do with, this doesn't have to do with reproduction. This is just cell replication. So let's say our skin cells, we shed lots of skin cells all the time. So our skin cells replicate. They make new copies of themselves. And to do that, what happens is you have this DNA molecule. You have, what does it mean when there's ASE? Enzyme, exactly. So the enzyme unzips the double-stranded helix, right? So you end up with different sides. So now you have two new molecules, and then you start attaching the complementary base to each new strand. And so you end up with two chromosomes, two strands of DNA, not just one and two. So there's a parental strand, which are on the, in this case we'll say on the outside. They were the ones that were split apart up here, and then there was a new strand, which is the complement to the parental strand. And so these are the different base pairs: C, T, G, C, A, right? Oh, I know what I want to do. God. Okay. Let's see. Oh, that goes there. Okay. So, now, DNA, before kind of molecular biology and being able to do stuff with DNA, we just knew that DNA coded for certain genes, certain proteins, right? It turns out once we could actually read what they coded for, and then we could collect it, and then people's DNA is different. We all have, there's 3 billion bases, right? So each of us has certain bases that other people don't have. And you've probably heard of a genetic fingerprint, which is where, let's say, you're looking for, trying to identify paternity or plane crash victims or murderers or someone who's been murdered, right? And you can do it by getting a DNA fingerprint from each person. You just need one cell or a few. And then what would happen is something that's called, well, let's look at this. So, cells, we leave cells all over the place. So you can oftentimes harvest DNA. Now, in the past, you needed lots of DNA, but now you can actually amplify it and get a whole millions and millions and millions of strands. Okay? So, and that's what this is. It's called polymerase chain reaction, where you're actually taking some DNA, making it unwind, and then building complementary strands, and then you can do that over and over and over and over again. Okay? So you have these primers that are kind of like signposts, and then inside are the genes, right? So this is the actual genes that are being coded, or that code things, or the stuff that you're actually trying to, match. And nowadays, they can just take actually one cell, and they can amplify, is what it's called, into many, 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 many strands of DNA. Okay. So this is kind of how it goes. You have a DNA. Scientists can amplify using PCR, so you got 2 copies, and then 2 copies. You got 4, and so on, until each cycle the amount of DNA doubles, like an exponential mathematics algorithm. And as I mentioned, not all of our DNA codes for proteins. There's a lot of it that's very, very variable. Okay? That means even though it doesn't code for anything, you might have a C, one person might have a C in one place, another person might have an A in the same place, right? And if you did that and you say, okay, let's say I want to find out who this person is. If you just had one of those places, you wouldn't be able to tell. You'd have one out of 4 chances, right? Because there's 4 different bases. But they actually have picked like 13 of these variable regions. And so the odds of having the same base at each of these regions being identical becomes vanishingly small. You might, at the first place over on the left, you might both have a T, but on the next one, you might have a T and the other person might have a C. Okay? So to get all 13 is like coming up heads when you flip a coin 13 different times. But it's even worse than that because remember, you've got 4 bases instead of just heads and tails. Okay, so instead of actually each base as a difference, we do these repeats. These are variable, and it's actually short tandem repeats, STRs, and they usually consist of 3 or 4 bases long. Okay, and in this region, so instead of just saying, oh, this person has a C here or an A here, it's actually one person might have 1, 2, 3, 4, 5, 6, 7, 8. That's 8, right? And then you might have another one here, where you have T, A, G, C, T, A, G, C, and you only have 2. Okay, so that's how they actually do it. They're changing now, though. They're now going to single base pair substitutions, and they're doing more of them. So, like the chances of the same person having the same tandem repeats or the same exact bases, is like 1 in a trillion. Very, very, very small. Okay. So what you do is, of course, you amplify this using PCR. You attach the DNA sequences to either side of the repeats. And these regions on either side, they don't vary. They're like signposts. They're always the same. And then those primers, everything in the repeat region, they can amplify into many, 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 many copies. Okay. So remember, every person inherits 2 copies, one from mom and one from dad. Mom and dad might have the same number of repeats, or they might have different numbers. Dad might have 8, mom might have 13. Okay. Now, and then what this is these different people up on the top. And as I mentioned, each person gets one from mom and one from dad. So, this person right here has one from mom and one from dad. This person here, they both have the same number. Okay? This person here, one from mom, let's say, which we did on which, and one from dad. One from mom, and then this one, same, right? Okay, and if you have like these 13 different sections of repeats, it's very small chance of actually having the same number for different people. However, because descendants inherit different numbers of repeats from either mom or dad, you can often identify let's say, whose child is this? Or brothers and sisters, because remember, brothers and sisters are related by half. So they're probably going to end up with about half the repeats are the same. Okay. Now, depending on the repeats, how many repeats there are, some are going to be longer strands and some are going to be shorter, right? So if someone only has 3 repeats in this one area versus 8, the 8th strand one is going to be longer. It's also going to be heavier. We have more bases there, 8 versus 3. So you might want to find out, and you look at a person's, if we actually back up a second, okay? And so what we do is we actually put these fragments in this gel. We put an electrical charge on it, and the fragments, depending on how long they are, move at different speeds. So a light fragment is going to, this is a smaller fragment, right here. That means it's, let's say, only 3 bases long, and it's going to move further than this larger fragment up here, which is much slower. And then everything is moving from negative to positive. So if we look back, we have one, we put the DNA samples. Usually in the first well, you put in a sample that you know already. It's kind of you already know exactly how many repeats there are and everything, right? And then you apply an electrical charge. The DNA fragments move through the gel at different speeds. You let it run for a certain amount of time, and then you stop it and you get a gel with these 2 different fragments, because each one for each parent, and that's your fingerprint for that one area. Okay, so here we are again. Person 1 has a fragment this long, and it's the same actual fragment at that place. Mom and Dad were pretty identical there. The next one, person 2, of course, Mom might have had a shorter fragment. Here's Dad, and here's Mom. Okay, so you can see how each person has a different fingerprint. This is just one spot out of 13. Okay, so which STR is smaller? The bottom or the top? Smaller. Top. Okay. Smaller, less weight, right? So it moves faster. Bigger, moves more slowly, so it's at the top. And we're going, I should have probably put it this way, we're going from negative to positive. Okay? So, just remember, anything that's up near the top, that means it's smaller compared to something down further in the bottom. Okay. So, who's not the father? Go to the next slide. Okay. Now remember, mom and baby, they both have the same base here. In other words, the baby probably got it from mom. The baby's father has it here, so we want to know which individual is not the father. Exactly. So obviously neither the baby or mom has an allele up here, or actually an allele down here. Remember, with two people, you've got four different outcomes. You can have four different outcomes, which we'll look at in a second. Oh, so the more STRs that you test, the more you can narrow down the possibilities of who a person is. So, like I said, I think there's 13 that we use, but actually there's more now. So things are changing so that it's a better resolution. But you have a variable region on chromosome 2, a variable region on chromosome 3, on 4, 2 on 5, 7, 8, and then you also have a variable region on the X chromosome and the Y chromosome, which is the sex chromosomes. So you can obviously tell right away who's female and who's male, because females have two X's, so if there's no Y chromosome, in other words that variable region, then you can figure out it's not a male. Okay, so who is the... Huh? Mm-hmm. They're baby. And mom's right there, and dad's right there. Okay. So a DNA profile now is 13 STR markers or more, actually. And the probability of two unrelated people sharing a DNA profile at all 13 spots is, what is that? A billion? Yeah, 1 billion. Actually, it's better now. It's not just 13. So, if DNA evidence does not match the person, then you can rule that person out. So, let's say you have an air disaster and you want to find out with body parts who's who. You can eliminate who's who, or the same thing with a victim or, let's say, a perpetrator. In fact, obviously, if you're not the suspect, or you're not the person who did it, it might be good to give them a DNA sample so that they rule you out, unless they're a bunch of course, crooked cops. They take your DNA and then they plant it all over the place and frame you. It does happen. So what could the baby's STR pattern look like? So we'll say baby 1, 2, 3, 4. Okay, with baby 1, we can have, here's mom, and we'll say, here's dad. Let's say baby 2, here's dad, and here's mom again, same one. Baby 3, we'll say, here's mom down here, and dad could be up here. And then baby 4, dad could be down there, and again, mom could be here. Okay, so there's going to be one of 4 outcomes, just because you have a mom and a dad. And that's what we just did. Okay, some things are not possible, obviously. 3 can't do it. 4 can't do it. Okay? Now, there are some organisms that are polyploid. Like there are some plants that they've duplicated chromosomes, and so instead of being diploid, one from each, they actually have 2 from, let's say, each parent. So they're tetraploids. But for people, unless you have Down syndrome, you have 2, always 2. Okay. Is that it? Oh, okay, good. Okay. I think what I'll do is let me